Good morning. I thank you, first of all, to our Katip Çelebi University colleagues that they are visiting us today, and especially Ibrahim Prim Hocam. We are working on the same field, and today we are sharing our today's guest with them because he has been supported by TÜBİTAK project and here in Izmir with us. And I hopefully today we will listen to him and see how we can pursue our common goals and how we can work together. And Dr. Simon Rayner is a professor in bioinformatics in the Department of Medical Genetics, Oslo University. And with his PhD in computational solid-state physics, then he continued from physics to the life sciences, and he worked in the Human Genome Project. And he has academic and industry positions in UK, US, China, Denmark, and Norway, world tour, and now currently he's working in Oslo University since 10 years. And currently his work focuses on the understanding of how biological systems evolve under external influences and translating scientific findings into the application with the social and patient benefits, of course. And today he will share his experience in Norway and also what they are doing currently in the medical genetics department in Norway. Thank you, Simon, for visiting us, and we will also later continue how we are doing in IBG and in a translational way, and what we are doing in RareBoost rare disease project. Thank you.
So thank you for that kind introduction, and thank you for bringing me to Izmir and have the chance to meet so many interesting people.
Along with that.
Oh, okay. Yeah, I once gave a talk in Hong Kong, and I spent the whole talk like this because the microphone was too low. And also, I'm told that I have to stay in front of the podium because of the camera, which is like I'm the sort of person who loves walking around when I present. So I would do my absolute best to stay in front of the podium, but I apologize to anybody on camera if I disappear. It's because I've forgotten, and I will come back soon. Already from what I've heard from the introduction about the center, it seems there is a huge amount of overlapping interest in what we do. I am going to try and find the pointer. Oh, he said point over here. You said point here and then use this to change. Yes, okay, I've got it. So I am at Oslo University Hospital. And I have a dual position at the hospital and at the University of Oslo. I'm within the Department of Medical Genetics. That's the wrong. Yeah, I'm in the Department of Medical Genetics. So pretty much it's the thing you're doing here. We do both basic research and we do genetic diagnostic services. So as part of this, we house the Norwegian Sequencing Center, which is a national high throughput sequencing facility. We have state-of-the-art sequences of a range of different platforms and very high throughput. And because Norway is a small country, it's 5.5 million, we actually offer genetic diagnostics as a general service to anybody. So you can go to visit your doctor, that you're sick, and the doctor may end up suspecting, well, there could be a genetic component here. So they can order whole genome sequencing, for example, for the child and the parents, and also other family members, so they can build up a genetic profile. So we have this bioinformatics core that's 14 bioinformaticians, and these bioinformaticians, they handle data analysis, but also importantly, they set up the reference databases and they do report generation for the clinicians. So the clinicians have a report that they can give to the patients. Equally important is they provide genetic counseling. So you end up with a report saying we found these mutations, and then you have to understand, well, what is the significance? What is the risk associated with the mutations? What does it mean for my health? And also, there's a quality control group that ensures that all processes meet international standards. At the same time, we also perform basic research into the mechanisms of genetic disease and try to develop diagnostic methods. So this is the research groups. They generally focus on specific aspects of different types of genetic disease, cancer, rare brain diseases, psychiatry. And then there's my group, which is looking at system evolution. So what do I mean when I talk about evolution? So my basic research question is, how do systems evolve in response to external influences? So the obvious example is, if you're looking at a pathogen, how a pathogen evolves over the course of a pandemic. For example, with COVID, in the early stages of the pandemic, it had a long incubation time that allowed it to spread globally. Once it had become established globally, the incubation time shrunk. And you also find mutations as a consequence of the evolution of new strains after vaccination. But also you can find this in scientific publishing as well. If what we study is influenced by how we're evaluated. So we're all seeking that Nature publication, and we want to get funding. And in order to do that, there's certain key areas that you have to study. For example, if you're looking at HIV or influenza, those are highly studied pathogens, and you have a better chance of getting a high impact publication for one of those than if you're looking at something like Japanese encephalitis virus. I look at cancer as well, but more from a different angle of looking at game theory. So if you go ahead and treat a patient, typically you're giving them a drug and you're looking for maximum tolerable dose. We're looking at, if you actually provide a patient with 2 drugs, how those drugs play off against each other in the presence of the tumor. And this could be modeled using game theory. And then I'm looking at the non-coding genome and ethnicity. And then I'm also looking at how do we tie this all together? Because when in the work that we do, specifically in the work we do here, we're bringing along high-dimensional, highly diverse data sets, and we're trying to integrate them to do modeling. So how we set up data sharing solutions is also important. But as I said, what I'm going to talk about today is not the non-coding genome and ethnicity and some of the work that we're doing. So one of the common ways of studying is using Genome Wide Association Studies. And this is something that's been widely used. There's increasingly large GWAS studies that are trying to identify thousands of polymorphisms associated with hundreds of common diseases. When they first started doing GWAS studies, there was the expectation that they would find monogenic disorders. And the standard example, the classic example is cystic fibrosis, where they found a mutation in the CFTR gene. And that having 2 copies of this mutation meant you carried a very high risk for cystic fibrosis. And the idea was that, or the hope was that every disease they look at, they would find key mutations where it would be very easy to come up with a diagnosis. And this is something that within our department, Arvid Frenger, this is a recent publication by him where he found a variant in this gene could explain this particular rare disease. But the reality is that most disorders are polygenic. And so to try and address this, people now come up with gene panels. For example, to identify risk of breast cancer, there's this gene panel of 17 genes. We also do one for for heart disease, where we have a gene panel of multiple genes that we sequence to look for the presence of mutations, and then go ahead and try and estimate disease risk based on the presence of these mutations. But one thing that's generally overlooked in these studies is the issue of genetic diversity. And as this paper pointed out, the majority of Genome Wide Association Studies have been performed in Europeans. And this actually has a lot to do with, not so much because they don't care about other populations, but just generally it's easier to build up cohorts from European populations. There's easier communication, it's easier to travel. So trying to go and build up cohorts in Africa, for example, requires a lot more effort just from communication issues. But nevertheless, there's a need to study more diverse populations. For example, with cystic fibrosis, I talked about this mutation at position 508. That only accounts for about 29% of cases in people of African ancestry. There's another mutation at position 3120 in the same gene that, depending on your ancestry, accounts for 15 to 65% cases. So if you're doing a screening based on this and you have somebody from African ancestry, you're not going to give a correct diagnosis. And this is relevant because, for example, in Norway, we're a small population, but since the 1970s, there's been a rapid increase in immigration. And now 20% of the population are immigrants of diverse background. And some of the largest components is from Asia, including Turkey. So there's a large Turkish population inside Oslo. So you have to take this into account. If we want to do these genetic diversity studies, genetic diagnostic studies, we have to take into account population variation as well. And as a consequence, there has been a large number of national genome studies initiated where people have national genome projects. Last time I checked, there were about 50 different populations. One of the most significant is the H3 Africa project, where they've started trying to sequence populations from across the African continent. This shows different populations that have been sequenced in different projects. And there's clear separation. They've identified more than 3 million novel variants in the H3 Africa project. And by looking at those variants, there's clear delineation of these populations across the first two principal components that correspond to distinct geographical regions. However, the point is that most of these variants occur outside coding regions. The other thing I want to bring into this presentation is about type 2 diabetes. This is an important health concern. In recent decades, there's been a rapid increase in the number of cases. Now it's estimated there's more than 550 million people currently living with diabetes, of which more than 90% carry type 2 diabetes. But there's a growing awareness that there's a risk by ethnicity. People of East and Southeast Asian descent are more likely to develop type 2 diabetes at younger ages and with lower body mass index. And Asian Indians are 3 times more likely to develop gestational diabetes mellitus. So in Norway, my group's been fortunate to be able to work with this study cohort of Norwegian women who develop GDM. And this is divided between South Asian and Nordic women. So we can investigate these patients after they recover from GDM and to see whether they're normal glycemia or they're pre-diabetic, and according to if they're South Asian or Norwegian. And this shows an investigation of insulin tissue, adipose resistant for participants in this group. And I'm sorry, I'll stand here. Over here, there is, this shows response for the 4 different groups. The lower one shows normal glycemia Nordic, so they have the lowest resistance. The next curve up shows South Asian who are normal glycemic, but their curve is almost identical to the curve for the Nordic women who are pre-diabetic. So this means that the Asian women, despite having recovered from GDM, are carrying the same level of insulin resistance as women who are pre-diabetic and of Nordic descent. So there's this inherent risk that they're carrying. And there's been a number of high-profile studies where they've tried to integrate multiple type 2 diabetes studies from across different populations to try and build the profile up of how different cardiometabolic phenotypes associated with type 2 diabetes patients can be broken down into different biological processes. So we've got beta cell function, glycemic, body fat, and there's different SNPs associated with I got this back to front. Each of these phenotypes can be associated with different clusters of SNPs. So there's what you're seeing as a clinical phenotype can be understood in the context of the collection of different single nucleotide polymorphisms. So it's showing the important genetic component. And there was also the study that was published recently looking at type 2 diabetes in South Asians. And they show that there's distinct biological processes with insulin deficiency and lipodystrophy. This is why I never studied medicine. I can't say the words. Unfavorable fat distribution. So associated with this high risk of developing type 2 diabetes are particular biological processes. So while all these studies provide valuable insight into genetic changes that are occurring during onset of type 2 diabetes, they're focusing on changes that occur in the coding regions of the genome. And this only accounts for 2% of the genome. So they're looking at key SNPs or SNPs in key gene loci or changes in transcription profiles that occur during onset of type 2 diabetes. And as I said, in H3 Africa, there's 3 million SNPs, many of which, or the majority of which, are occurring in non-coding regions. So this is what I became interested in. There are many studies, both on non-coding regions and on diversity, but they're small scale. So this is showing epigenetic alterations in Chinese, Tibetan, and Han populations. So it's a small study on a small study group looking at specific changes in genes. It's not a genome-wide screening. What we obviously we can't look at the whole non-coding genome in one go. So we started by looking at microRNAs. MicroRNAs are small non-coding RNA molecules, about 22 nucleotides, but as something happened on the Zoom workspace. There's a message coming up on Zoom workplace, like maybe it dropped the connection.
It's okay.
Is that okay?
It's okay. Yeah.
Okay. My Turkish isn't what it used to be. Okay. So they play an important role in gene regulation. They bind to the 3' UTR of genes, causing post-transcriptional regulation of genes. Their role in type 2 diabetes has been studied widely. They've identified particular microRNAs that are dysregulated in the presence of type 2 diabetes, and it's correlated with their predicted target. So microRNA binds to target, and if it's upregulated, then it pulls down its target. So this is the basic model, that you have a microRNA is transcribed for one genome location, and it goes ahead and binds to the 3' UTR of another microRNA that was transcribed from a different region of the genome. But the important thing here is that the microRNA does not perfectly bind to the mRNA. It's a very noisy binding. So instead of saying it's very difficult to accurately predict where a microRNA is going to bind. Last time I checked, there's 129,000 microRNA-related publications in PubMed. But these studies focus on identifying dysregulated microRNAs between conditions and predicting their gene targets. So they're still focusing. You're finding this microRNA, this non-coding feature, is dysregulated, but you're still focusing on the coding region. The reality is that the microRNA targeting process is actually more complicated. So this is what I just talked about. A microRNA binds a 3' UTR. In reality, one microRNA, due to this binding process, can target multiple mRNAs. And at the same time, one mRNA can be targeted by multiple microRNAs. So what you end up with is complex mRNA, microRNA networks where you have this high connectivity between a microRNA and its mRNA. So how can we investigate these networks on a genome-wide basis? So rather than looking at a small number of microRNAs, we want to do this in a global way. So there's a whole question of microRNA target prediction. There are target prediction tools, but things like target scan, you can only do this online. So you can say, here's my gene, and it will give you a list of predictions. But we have 20,000 genes. And also another point that I'm not going to get into too much detail, but when people train these models, they say to the model, well, the seed region is really important, so look out for this. So rather than letting the model decide for itself, it's trying to sort of push the model. I'm moving again, sorry. It's trying to push the model into the direction you think is important. So we kind of took an unbiased approach and said, this microRNA targets this 3' UTR, you go and figure out what's important. So our problem again was insufficient training data. We had lots of positive data. We didn't have so much negative data. So we had a very imbalanced data set, which limited our model performance. But nevertheless, it means that we can do a high throughput system-wide investigation of microRNAs, target networks. I say we outperform all of the target prediction tools, but as I said before, it's still not great performance. No microRNA target prediction tools work outstandingly well because of the limited amount of data that's available. And then there's the issue of data integration that I talked about briefly in the introduction. We're trying to use all these different data types. We want to look at 3' UTRs, so we're using the Ensemble database. We're looking at microRNAs, so we're using MIRBase. We're interested in how microRNAs are conserved across species, so we're looking at RFAM. And we're also interested in what 3' UTRs and microRNAs occur in what pathways, so we use the Reactome database. And then we need population data, so we use the 1000 Genomes project, which actually is now 4,978 samples across 26 different subpopulations and 5 superpopulations referencing the continents where the samples were collected. And this information is in a variable call format. And so we bring this data in, and we have a couple of tools, Mirror for target prediction and this PyNonc that kind of brings this data together. And I'll come back to this in a little bit. So this does all the processing in a very automated way. And from this, I want to just present some of the basic characterization that we observed of the 1000 genomes in terms of microRNAs and the 3' UTRs that they target. So this shows all populations, and it's showing that more important 3' UTRs, and by more important, we're looking at the ones that occur more frequently in the Reactome pathways. The Reactome database contains 2,400 and something pathways. So here you've got 3' UTRs that occur in 500 different Reactome pathways, and these contain small number of population SNPs. Here, the 3' UTRs that are only involved in 1 or 2 pathways contain large numbers of population-related SNPs. And we see the same pattern here for microRNAs. If a microRNA is highly conserved, so you see multiple species, you see it present in multiple species, it contains small numbers of SNPs. If the microRNA is unique to a particular species like human, then you see many more population mutations accumulating. And we also see, as we expect, relative to the standard reference database, we see larger numbers of variants in African populations, both for microRNAs and 3' UTRs. So this is the this is just a randomly selected signaling pathway. I don't remember why we selected that one, but this is the interaction pathway. This is showing the target predictions of all the microRNAs and which 3' UTRs they target. The red lines show experimentally verified targets. But then how do you characterize these? We can see that there's a large number of population-specific mutations occurring in this target regulation network. So this is just showing for African, Caribbean SNPs. And for example, there's this microRNA. If we go ahead and look at this, we can look at the connectivity. So how many connections does each node have? I.e., how many 3' UTRs does each microRNA target? And this shows this one. It says this microRNA fine-tunes growth and development and has a role in cancer neural repair. And it turns out this one that has a role in cancer is one of the most highly connected microRNAs in this pathway. So microRNA, and we haven't done this for every single pathway, but each pathway we looked at, because we're kind of just inspecting our results at the moment. Many of or the majority of the highly connected microRNAs are ones that have roles in disease like cancer. And the weaker ones that contain more SNPs, population-specific SNPs, are weakly connected. So again, there seems to be this pattern that population SNPs are kind of fine-tuning networks. They're not doing massive perturbations to the network. They're just kind of giving some sort of tweak in behavior of the targeting. But another interesting thing is that there's families of highly similar microRNAs, of which there's this one, and this shows the microRNAs that are participating in this part of the network, not this one, this one. So that I think it's 29 highly similar microRNAs that belong to the 548J family. And if we look at these microRNAs, how single nucleotide polymorphisms are accumulating in populations, we see something like this. This is showing the 26 different populations in the 1,000 genomes. And these are the different microRNAs in the 548 family. And every population has a slightly different mutation pattern. So they have different, you can actually identify a particular population based on its mutation pattern of this family of microRNAs. And we can even identify two of the populations are North Han Chinese and South Han Chinese. And even there, we can distinguish between these two quite similar populations simply based on the presence or absence of mutations in this small family of 29 microRNAs. But so far, I've just been talking about the effect for one trait, in this case, population, in one family of microRNAs. How do we examine all 3' UTRs and all microRNAs for different traits? So typically what you would do, for example, if we go back to the world of virology, what you do is you collect a set of different samples from around where an outbreak is taking place. You'd go ahead and you'd sequence these, and then you'd go ahead and build a tree. So for example, this is showing a result from a study I did when I was living in China, looking at RareBoost strains. You can see a very clear lineage replacement event. So in 2011, there was similar amounts of these two lineages. By 2 years later, this lineage had displaced this one. And we could also see geographical patterns. We could see how the virus was spreading across China and was responsible for new cases in the epizootic. So within 2 years, we could see that China 1 was a dominant lineage. So the way we did that was we identified this lineage shift by looking at one protein, the glycoprotein, plotting a tree and looking at the tree pattern. This was simple because RareBoost is a small virus. It's about 11,000 nucleotides. It's only got 5 main proteins. And we knew which protein to look at. The glycoprotein, it's a surface protein. So it's a rapidly evolving protein. Oops. But what about the larger genomes that we're talking about? We've got 3 main projects we're looking at. One is this human cytomegalovirus. That's an important virus that causes birth defects and neurological problems. It's a DNA virus. It's 22,000 nucleotides. It has 117 proteins. Malaria, 23 megabases, 5,500 proteins. Homo sapien, 3.3 gigabases and 20,000 proteins. You can't plot a tree for every single protein. Start looking and go, oh, no, no. And also our data sets. For CMV, we're currently working with 410 genome sequences. Malaria is 67. Human, we're working with 3,500. So even if we look at one tree, it's difficult to see any pattern. This shows a single gene for CMV for one protein. And how do you identify if there's any clustering or any interesting clustering of CMV? We're interested in, do we have, we're looking at clinical strains versus lab strains or drug resistant versus drug naive. Looking at these trees, there's nothing you can do. And also, in the context of the analysis of the human genome, we're interested in looking at how individual reactor and pathways. I looked at this ERBB2 signaling pathway. So we don't just want to look for patterns across genes and microRNAs. We also want to look at patterns across signaling pathways. So basically, what we want to do is find interesting trees. So this is an example of an interesting tree. There's 3 different populations: Finland, Japan, and Nigeria. The Finnish samples are grouped up here. The Japanese samples are in this cluster. Nigerian samples are sort of clustered here. This one Nigerian sample is not quite in the right place. So this is an interesting tree. There's strong clustering by sample feature, in this case, geographical location. This is a boring tree. Everything's mixed up. There's no clear selection pressure acting on these sequences. But these are also interesting trees. The Japan and Finnish ones are not clustering, but the Nigerian ones are still clustered together. So we're also interested in the ones where you have a single feature is clustered in the tree. And this is where this program, Willow J, comes in, that we developed. It was developed by a master student of mine, actually. And to identify the interesting trees, Willow calculates a quantity called the group diversity ratio, which is defined as the mean intergroup pairwise distance and the mean divided by the mean intergroup pairwise distance. And I thought I'd try and explain this. I'm not going to go into too much detail, but I thought just to try and give you some kind of idea about what this means. This shows a simple tree. There's 7 samples in this tree. The blue ones are fairly well clustered, but there's got this one blue one over here. The green ones are actually quite well clustered. They're all in a single clade. So we've got 7 samples, 2 groups, A, B, C, G, D, E, E, F. So we can go ahead and try and calculate this distance. And if we look at the distance matrix, we can see the distance between each of the pairs of green samples is less than the blue-green distance. So we've got 0.5, 0.5, 0.5, and these ones are identical because it's just a square distance matrix. So 0.5 is always less than the distance is 0.8, 0.8, 0.7. So you can see right away the mean distances between the green samples is going to be less than it is between the blue and the green samples. So if you want to calculate the intergroup distance, that's the group with the distances between all samples in the same group, you have to calculate AB, AC, BC, AG, BG, CC. So it's AB is the distance from here to here, AC is the distance from here to here, BC is the distance from here to here. That's 0.2, 0.4, 0.4. And then this is a larger distance. So this got it all up the tree, back down the other way. That's 0.8, 0.8, 0.8. So you add those all up, and we did 6 pairwise distances. So the mean distance is 0.67. We can do the same for the green group. In this case, we've got 3 distances, DE, DF, EF. So you've got 0.4, 0.5, 0.5. So the mean intergroup distance is 0.467. Then we have to go and calculate the intergroup pairwise distances. So this is the distance between all pairs in the group you're looking at, which is the blue group, and all the ones in the green group. So we have only got 2 here, so it's a simple case. So then we've got all the way up from A down to D, A through E, A through F, and we do it in each case. And what we end up with is 12 different pairwise distances and an average of 0.7. And of course, we've only got two groups. So if we do the same thing for the green-blue, we're going to get the same value of 0.7. And so finally, the mean intergroup pairwise distance is the average of the blue intergroup distance and the green intergroup distance, which is, I don't know what that is, 0.438, I think. And for we can do it for so we end up with a value of 0.738, and we do it for the individual groups as well. We get 0.67 and 0.667. So there's the GDR value of the green group is lower than the GDR group. And that makes sense because if we go back to the original tree, we can see that these are more tightly clustered than these because this 1G, that's what's pushing the GDR value up, so that there's less clustering. But nevertheless, this tree shows good, what we call phylogenetic structure, that both samples are kind of clustered by their feature. So go back to the tree. So that means that if we have a distance matrix, then we can go ahead and we can calculate how well clustered the samples are for that distance matrix. But to get a distance matrix, we need a phylogenetic tree. To get a phylogenetic tree, we need an alignment. To get an alignment, we need FASTA sequences. So how do we get the FASTA sequences? It depends what we're doing. For the 1000 Genomes project, we have to pass out the VCF information, and that means, and from the VCF information, we have to go ahead and use the ensemble information to get the sequences, and then we have to update the sequences. If we're working with something like CMV, there's no single standard reference because it's a virus that's rapidly evolving. So what you have to do is say, well, okay, this is going to be my reference genome, and then you've got to just you have to annotate that and then get out the features of interest. So what we do is we have a raw sequence, we have a gene bank file. The gene bank file contains the position of all the interesting features. So there's 170 genes in the gene bank file. So we generate 170 BLAST databases, and then we blast each of the 170 BLAST databases against the query sequences. So we have 170 times 410, and then we have a parser that brings it out to generate the FASTA sequences. So the thing is, from both these routes, we end up with a set of FASTA sequences to build the alignment, to build the phylogenetic tree, to build the distance matrix, to run Willow. So that means that we have a fully automated pipeline that can either take a VCF file or that can take a reference genome with position annotation, and then we can run through and investigate how well each gene, or in our case, each non-coding feature is clustered according to the feature of interest. In our feature is the geographical location of the sequences, the population. So what do these results look like? This shows a curve of GDR values for 8,600 genes. There's a small number of genes with low GDR values. So these are the ones that have very strong population structure. It means that the sequences are clustered according to their geographical origin. And the rest of them, there's no geographical structure. However, this is the plot just for the trees. This is from when we originally wrote the program, before we realized that we were also interested in trees where you could have a single population could be clustered and all the other populations could be randomly distributed. And this is sort of preliminary results that we're still trying to process. If we look in the context of looking at reactome pathways, and we look at type 2 diabetes, the hypoxia-inducible factor pathway plays a key role in type 2 diabetes, because you see impaired activation of the pathway, which weakens response to hypoxia, leading to downregulation of its downstream target genes, and it leads to things like foot ulcers in type 2 diabetes patients. If we look at the results, this shows a summary of the GDR values that were calculated for the 11 for the genes that are in the that are part of the H1F pathway according to the Reactome database. This is color coded, so values that are red have low GDR values, and it means that there's some kind of selective pressure acting on the 3' UTR sequences in that particular population, which means they have distinct sequences that are unique to that population. So we can see in East Asian populations, there's many genes in this pathway that have some sequence features that are unique to these populations. And that means if they have unique features, it means it's going to affect their target network, because different sequences means different targeting by microRNAs. In Africa, where there's also a risk-associated genetic risk with diabetes, it seems to be a single gene. But in European populations and American populations, there doesn't seem to be such a strong signal. Although, interestingly, there is this one population, PEL, that seems to have strong genetic signal. And if I go and look this one up, it's Peruvians living in the Andes at high altitude. So there's some kind of adaptation for high altitude for oxygen conversion. So with this approach, we can perform genome-wide screening of non-coding regions. So we now, in the first version of this WillowJ program, we wrote it in Python, and we realized it was going to take us 6 months just to do the calculations on all the genes. We rewrote that in Java, and it runs 1,000 times faster now. So now we can do all 3' UTRs, all microRNAs, all target, and we can do bootstrapping to estimate statistical reliability of our results. So we can look at how do SNPs accumulate in features by populations, how do they accumulate in pathways by population, and also how does the network structure vary in pathways? And this is what we're trying to do right now. And the next steps, we've actually collected our own cohort of pre-diabetic patients with matched controls from Addis Ababa. But then rather than just focusing on what gene expression changes occur between the two groups, we've done whole genome sequencing so we can identify population-specific SNPs. We can do epigenetic changes so we can find where epigenetic markers are present. And again, this is usually done in the context of coding regions, but this can also impact non-coding as well. So if you have an epigenetic mark upstream of where a microRNA is transcribed, that can shut down the expression of that microRNA. And then we can see the impact of these mutations and epigenetic changes by looking at gene expression in RNA-Seq and small RNA-Seq to get changes in microRNA profiles. And we haven't found anyone that's actually put all 4 platforms together. You always get 1 or 2, but never all of them. And so what this means is, from a network perspective, we can see how population SNPs modify a regulation network locally, and how epigenetic changes can shut down an entire region of the network. We can have a much more significant perturbation on the network. And then what we want to try and do is apply food web theory to this. And food web theory is a model that tries to describe an ecosystem containing S species. And you have a community matrix to describe the interaction between these species. So where element ij describes the effect of species J as on species I. And from these theoretical models, you can talk about predator-prey, mutualistic, competitive, and so on. And they find that predator-prey interactions are stabilizing, whereas mutualistic competitive are destabilizing. And the stability is dependent on its connectance. So in our implementation, we view that microRNAs are the prey. They prey on 3' UTRs. And if we have a situation like this, where you have an epigenetic change, that has a big impact on connectivity. And the population SNPs are also going to have some effect on connectivity. So you have some implicit difference in the connectivity of specific networks between 2 different populations. And in that case, having an epigenetic change can have a much more significant effect on a particular population. And the hypothesis is that as you accumulate epigenetic changes, that leads to kind of a tipping point where the connectivity of the network collapses. And so this is what we're trying to use fluid web theory for. An important part of what we do as well is training. There's been a lot of talk about helicopter science, where these rich European countries come in and say, oh, thank you very much for that data. And this happened a lot with like Ebola. In Ebola, there was a big global outbreak of Ebola. A lot of European labs came in, collected samples, took the samples out, sequenced them, and went ahead and published it in Nature. And there was, in many cases, there was relatively limited acknowledgement of the roles played by the people in Africa. So for the last 3 years, we've been carrying out bioinformatics trainings in Africa, and so that people can actually be responsible for analyzing the data collected in their own countries. And so we're co-mentoring several students on master's project related to this work. And this particular picture was actually quite satisfying because we managed to get students from Ethiopia and Central West Africa into the group at the same time, so that everybody was actually working together. And it also seems to be a really nice way of bringing people together, because like any country, it's like if you're working in like the military, and when I was in China, I was in the Chinese Academy of Sciences, and the Chinese Academy of Sciences were like, well, we're the Chinese Academy of Sciences, and you're just a university. And then there's the military. It's like, well, we're not working with the universities, and we're not working with the Chinese Academy of Sciences. And there was a similar situation here, but what happened was, because people were suffering through this bioinformatics course, like you suffered through my bioinformatics course last week, it's like you shared the pain, and there was some sort of community that was built up. And this has happened now where these people, they don't really care what institute they're from. They're kind of friends now working on bioinformatics protocols. So in conclusion, I would claim that the non-coding genome is an under-researched region of the genome, has potential to produce new insight into disease mechanisms and genetic risk, and offering new diagnostic possibilities. But to do that, we need to adopt new perspectives that move beyond the gene-centric view of the genome. And with that, this is my group. This is their picture. This is showing the conflict between experimental and bioinformatics. The constant conflict exists in, I think, in any research environment. Thank you very much.